Hello, this is Dr. Rinarani Rai. Welcome to the second part of our bacterial staining discussion. Before starting, let us discuss about fixatives, mordants, and dyes. So what is fixation and fixative? Fixation is a preservation of biological tissues from decay due to autolysis. It terminates all ongoing biochemical reactions, thus increases the treated tissue's mechanical strength or stability. Fixatives denature proteins by coagulation, by forming additive compounds, or by a combination of these two. Some common fixatives are ethyl alcohol, formalin, acetic acid, chloroform, mercury chloride, chromic acid, picric acid, osmic acid, and others. Now, what is mordant? Some stains do not have natural affinity for the cells. So they are mixed with salts like oxides of aluminium or chromium to form insoluble coordination complex. This now binds with the target tissue with greater affinity. These are not stains, but used to stain to increase its binding capacity. Example, as we have seen in gram staining, that is a 40% of potassium iodide increases the binding of crystal violet with peptidoglycan of bacterial cell wall. So here we can see application of mordant increases the affinity of dye towards the tissue. Now what is dye? A dye is a colored substance that chemically binds to the substrate to which it is being applied. This distinguishes dyes from pigments, which do not chemically bind to the material they color. The dye is generally applied in an aqueous solution and may require a mordant to improve the fastness of the dye on the fiber. Dyes differ from pigments, which are finely ground solids dispersed in a liquid such as paint or ink or blended with other materials. Most dyes are organic compounds. That is, they contain carbon, whereas pigments may be inorganic compounds or organic compounds as well. Now we are coming to the first type of staining, that is endospore staining. So endospore is a dormant, tough, non-reproductive structure produced by a small number of bacteria. The primary function is to ensure the survival of a bacterium through periods of environmental stress. Endospores make bacteria very difficult to kill. So here we can see the blue colored endospores and the vegetative part is appearing in the pink color. So there are 6 types of endospores. The first one is oval, centrally placed but not distended. The second one, as we can see, it's oval but terminally placed, non-distended type. Third one is oval, terminal and distended. Fourth one is also oval but centrally placed and distended. Fifth one is spherical, terminally placed and distended. And sixth one is oval, lateral but distended. So bacterial spores have proven to be difficult to stain as they are not permeable to aqueous dye reagents. Endospore staining is particularly useful for identifying endospore forming bacterial pathogens such as Clostridium difficile. In 1922, Dorner published a method for staining these endospores. Schaefer and Fulton modified Dorner's method in 1933 to make the process faster. And this modified Dorner method of endospore staining uses malachite green dye and safranin. So how will we stain the endospore? First, after preparing the bacterial smear on the glass slide, we will apply the malachite green, and this is the primary stain. Then application of heat, where heat of steam is acting as a mordant. Next step is the application of water, and water is acting as a decolorizer to get rid of the excess stain. And lastly, the counter staining part, that is application of safranin, is done. Malachite green dye, that's alkaline with a pH of 11 to 11.2, and it's a water-soluble dye. It binds to the spore mildly. Now steamed heat is used, which softens the endospore and allow the penetration of the dye into the spore. Water is used as a decolorizing agent to wash away the malachite dye excess from vegetative forms. And lastly, the use of a counter stain, the safranin reagent, is also known as the secondary stain. It is to stain the vegetative forms of the underdeveloped vegetative forms. It can detect the presence of endospores in 6 genera of bacteria, where endospores appear as green and vegetative cells will appear in red color. So here we can see malachite green. This stain, they are having the positive charges around, and the bacterial cytoplasm, as we know, they are containing the negative charges allowed. Now endospores will appear as green dotted elliptical particles for malachite green dye, whereas vegetative cells will appear in pink red. Now, the next type of staining technique, which is known as acid-fast stain, that is a Ziehl-Neelsen technique. Acid fastness is a physical property that gives a bacterium the ability to resist decolorization by acids during staining procedures. Once the bacterium is stained, it cannot be decolorized using acids routinely used in the process. Ziehl-Neelsen staining is a bacteriological stain used to identify acid-fast organisms. Franz Ziehl and Frederick Nielsen, they have proposed this staining technique, which is being used to stain the genus Mycobacterium and also genus Nocardia. Here, the primary dye used is a carbol-fuchsin, and the counter stain is a methylene blue. Here we can see the methylene blue in the bluish tinge, and carbol-fuchsin is a red color, and acid alcohol is used as a decolorizer. First of all, the application of carbol-fuchsin, that is a primary stain, on the bacterial smear present on the glass slide. Here also, the application of heat, where steam is used as a mordant. Then application of acid alcohol, which is acting as a decolorizer. And finally, the application of methylene blue, which is acting as a counter stain. When the smear is stained with carbol-fuchsin, it solubilizes the lipoidal material present in the mycobacterial cell wall. By the application of heat, carbol-fuchsin further penetrates through lipoidal wall and enters into the cytoplasm, and cell appears red. Then the smear is decolorized with decolorizing agent, that is 3% hydrochloric acid in 95% of alcohol. But the acid-fast cells are resistant due to the presence of large amount of lipoidal material in their cell wall, which prevents the penetration of decolorizing solution. The non-acid-fast organism lack the lipoidal material in their cell wall, due to which they are easily decolorized, making the cells colorless. Now, some important things to remember. Mycobacterium tuberculosis is killed by sodium hypochlorite or the bleaching powder. So if the smear is not made from bleach-treated sputum, that is taken from some patient, alcohol fixation is a must, as it can kill the bacteria. Heat fixation cannot kill this Mycobacterium tuberculosis. And do not overheat the stain, because heat the stain until vapor just begins to rise 60 degrees centigrade. Overheat will spoil the bacterial smear. The bacteria is moving. It is moving through the help of flagella. So how can we stain the flagella, bacterial flagella? So flagella are too thin to be visualized using a bright field microscope with ordinary stains, such as the gram stain or a simple stain. A wet mount technique is used for staining bacterial flagella, and it is simple and useful when the number and arrangement of flagella are critical to the identification of species of motile bacteria. The staining procedures require the use of a mordant so that the stain adheres in layers to the flagella, allowing visualization. Flagella is colored or stained by Leaf-Sohn's method. This technique is used to visualize the presence and arrangement of flagella for the presumptive identification of motile bacterial species. A wet mount technique is used for staining bacterial flagella, and it is simple and useful when the number and arrangement of flagella are critical to the identification of species of motile bacteria. The staining procedures require the use of a mordant so that the stain adheres in layers of the flagella, allowing visualization. Now we are coming to the procedure of staining. First, grow the organism to be stained at room temperature on blood agar for 16 to 24 hours. Add a small drop of water to a microscope slide. Then dip a sterile inoculating loop into the sterile water. Touch the loop full of water to the colony margin briefly, which allows motile cells to swim into the droplet of water. Touch the loop full of motile cells to the drop of water on the slide. Agitating the loop in the droplet of water on the slide, it will cause the flagella to shear off the cell. Cover the faintly turbid drop of water on the slide with the cover sleeve. Now, a proper wet mount has barely enough liquid to fill the space under a cover slip, so small layer spaces around the edge are preferable. Examine the slide immediately under 40x to 50x for motile cells. If motile cells are not seen, do not proceed with the stain. If motile cells are seen, leave the slide at room temperature for 5 to 10 minutes. This allows the bacterial cells time to adhere either to the glass slide or to the cover slip. Gently apply 2 drops of RYU flagella stain, that is Remel Lenexa cancers, to the edge of the cover slip. The stain will follow by capillary action and mix with the cell suspension. Small air pockets around the edge of the wet mount are useful in aiding the capillary action. So after 5 to 10 minutes at room temperature, examine the cells for flagella. Cells with flagella may be observed at 100x, that is under oil immersion in the zone of optimum stain concentration, about halfway from the edge of the cover slip to the center of the mount. Focusing the microscope on the cells attached to the cover slip rather than on the cells attached to the slide facilitates visualization of the flagella. Now, what we will see, we can identify the presence or absence of flagella, number of flagella per cell, location of flagella per cell, that is whether it's peritrichous, lophotrichous or polar, and the amplitude of wavelengths, that is short or long. Here we can see the arrangement of flagella in different bacteria. And so this is all about the staining procedure, and thank you for your patient hearing.